Thank you, David. A fascinating subject, and I'm sure there'll be questions at the end and during the cocktail how this all plays out and the deep learning, big data, and blockchain technologies all coming together. We're now going to shift topics and subjects, and it is still about, of course, new thinking from outside of your industry, and how that can make a difference in revolutionizing vaccines. So I'm going to very soon welcome Benjamin Damien. Benjamin is Director of Business Development at Univercells. Benjamin holds a PhD in molecular biology. After a few years in fundamental research on vaccines and cancer, Benjamin oriented his career towards management and innovation. Benjamin created his first startup in 2003 and has so far created 5 companies and been CEO of 2 of them. So a serial entrepreneur, as we say. And since 2014, Benjamin works with Univercells as Director of Business Development, bringing his expertise of the biotechnology sector and his international network. So Benjamin will share with us how looking outside the industry for new thinking and insights to solve a problem can actually impact global health issues. So this is about changing the rules of manufacturing, of biomanufacturing, and changing the paradigm of vaccine manufacturing and availability. Benjamin has a passion for fostering innovation and entrepreneurship, as we can see from his career, especially with students and lecturers, and really encourage them to become entrepreneurs themselves. Please join me in welcoming Benjamin Damien.
Okay, thank you for the introduction and for making me sound more interesting than I'm probably really are. I'm indeed Benjamin Damien. I will quickly try to convince you that with innovation we can drastically reduce the cost of making vaccines. So I hope that here we are all convinced that vaccines are very important tools for healthcare. You see here the decrease. Is this working? No. The decrease of disease thanks to vaccination. So we are living, thanks to vaccination, to basically a disease-free world, at least for infectious disease. This is not all nice and peachy, because there's a few issues with it. For example, in the developing world, there's still a large portion of the population, especially children, that are not vaccinated, just usually by availability or cost. And a big proportion of those will die before the age of 5. There's also a problem of supply and availability. A lot of those vaccines are manufactured only by a couple of producers. So if anything happens to the factory, you get a lack of availability of the product. And there's also health crisis, if here Zika or Ebola or even pandemic flu, when suddenly the world needs a few millions of vaccines and you've out of capacity, you are just not able to produce enough of the product for the needs. So there's a real need for increased capacity of production and for cheaper vaccines. And I like to say that despite the world vaccine market being almost $50 billion, this is mostly in US, Europe, and Japan, and made by 4 companies. The rest of the world is basically still outside of that part of the market. So we should try to empower those poor countries to be able to produce their own vaccine for their own population. You see that this is a map of the availability of vaccines, which is strongly correlated with GDP and with the organization of the country. So the poorer, the less vaccine you're probably getting. So why are vaccines expensive, and why do we have limited capacity of production? Is that, well, for making vaccines, you basically need to grow cells and infect it with a virus that will kill the cell and grow. And you need for that a surface. The cells need to grow on a surface. So usually you need bottles. You cultivate your virus in a bottle, and if you need more virus, you take more bottles. You make more bigger bottles and so on, or more bottles. And of course we need millions of those. So you end up having huge factories with millions of bottles. Or even in some of them, we are still making vaccine in eggs. I think Sanofi is going every day through dozens of, tens of or thousands of eggs per day. There's still possibility to use reactors for making vaccines. So it's basically a big vessel in which you put your culture medium, and as the cells have to grow something, you put small beads, microscopic beads, and the cell will grow on the surface of the beads. And that's That kind of system is about 1,000 liters maximum, and we can reach a density of 1 million cells per ml. Keep that number in mind, and we'll come back to that later. So 1 million cells per ml. So we basically have two situations here. We have the old way with the small bottles that are used in big numbers, which is about 80% of the vaccine manufacturing. Of course, you can imagine that if you have to handle 1 million bottles for making your product, every time you have to, ask somebody to take the bottle, open the bottle, empty the medium, put a new one. So it's changed for having an infection or having a contamination 1 million times in every time you handle your batch. So there's a lot of chance of having problems with your production. And of course, you need huge factories because 1 million bottles factory is quite sizable. So it makes very expensive factories, and you need a lot of workforce for that. And usually you have a relatively limited capacity of production compared to the volume. And you have the lower one, which is the one using bioreactors, which are, of course, less manipulation, so it's much safer, but of course the factory are much, much more expensive. So expensive is not a problem if you are GSK or Pfizer or Merck. It is a problem if you are an emerging country or if you are a small player. So what are the barriers to entry? As I said, factory are extremely expensive. A vaccine factory costs over $100 million. The manufacturing process is complex, so you need a lot of workforce, well-trained, which is also difficult to get in emerging countries. Production is still based on batches, so basically you fill up your bioreactor, you harvest it, then you clarify your harvest, then you purify your harvest, and do that step by step, and then you start. You start again. It's like for the car industry. A century ago, you were building cars in a garage when people were working on the same car, and when it was finished, the car was going out. And then Ford came with the chain production. When you come up with the small parts in one side, you need to come up with the car on the other side, and that can be a continuous process of production. The system uses usually low-density manufacturing. I would say 1 million cells per ml. Meaningless number, but it's a relatively low number in terms of density. And low density means high cost per volume, so high cost per vaccine. And of course, there's a lot of regulatory issues and control, which is extremely complicated for manufacturing, and this prevents small players and big countries to come into the process. So we need innovation in manufacturing. What I've just shown you is not very innovative. It's just engineering your way out of a problem without changing the underlying principle of the production. And we could, for example, increase the density of the manufacturing, make more in a smaller volume, and chain the operation, make all the things in one go, like the Ford car factory, which is not the case at the moment. Simplify the QC, make smaller factories, and so on. So how could we do that? Yeah. So one of the steps has been taken, for example, by a bioreactor called iCellis, which is sold by Paul of Science, which is one of these small bioreactors is basically filled with fibers. So it's like a 3D matrix of plastic. And cells are adhering on that. So you basically have a very high density matrix of cells. We can reach, I think it's there, 40 or 200 million cells per ml. So it's 100 times more cells per volume that the bioreactor I showed you before. So you could use 100 times smaller bioreactor for the same kind of productivity, which is nice. So it's equivalent to 3,000 bottles that I showed you in one of the first slides. So in terms of simplification, it's already a huge step forward. You can do better. So in industry, in chemical industry, chaining and continuous process is normal. For metal production, in the good old days, you use a furnace. Basically, you dig a hole in the ground, you fill it with bricks, you put your metal ore in there and coal, you light it up, and then you end up with some metal that can work in a forge. But that's what we remade metal, cheap and usable, is using the blast furnace, which is a continuous process. You have a flame in there which is alimented by coal, and you put your metal on the top, and you end up with a continuous casting on the other side. And you do the same for petrol, you do the same for cars, you manufacture your phone in the same way. So continuous processing, it's normal in the industry, but it's not done usually in biotech. So we could imagine to have a process when every step of the manufacturing process of vaccine will be concentrated in one machine. And we start with the bioreactor you see on the top right, which is our development. It's the same principle of the iCellis. It's a small, simple drum with fibers in there. We can reach even higher density. We reach 240 million cells, so 240 times the first bioreactor. It's a very simple design. It's built like a washing machine drum, so it kind of spins, so it kind of homogenizes the whole thing. Of course, we have a very high density of cells, so the cell will eat all the product in the culture medium, so you need to replace it at some point. So you need to perfuse medium, you put new medium in there, and as you have putting new medium in, you have to take some liquid out, otherwise it will overflow. So you will take the waste out, but you will also take your product out. So you could imagine to connect your purification directly outside the bioreactor, and you will purify on the go your new product. And for that, as you are not dealing with 1,000 liters in one go, but a little volume per hour, you could use very small columns. And columns of purification are very expensive. So you save money by doing that. You basically create a system which is there on the right, which is about the size of a cupboard. It's 2 square meters. We can produce 40 million doses of vaccines per year, and it's extremely cheap to run and cheap to operate. We could even go one step further because, okay, you got that machine, you still need a factory, and factory expensive. What if you could put all that into a system which will require no factory? And that's what we're trying to do with the help of the Gates Foundation, is basically we've embedded all this perfusion system into an isolator, which is 3 meters on 2, which is basically your factory. So I won't say that you can use that into a garage, but it can at least simplify largely the design of this factory. We estimate that less than $10 million factory could produce 40 to 80 million doses of vaccines per year, which is about a fourth of the need in the world of polio vaccines, for example. So it's a huge kind of capacity of production. Reduce the cost of factory, increase safety. Safety for the product, no contamination, but safety for the operator. It cannot be infected because it's not directly in contact with the product. Safety for the environment. You can imagine that if that leaks into a river when you're producing vaccines for polio in India, you're going to kill thousands of people. That's limit the, of course, difficulties or risk of that. And of course, with a very simple process, simple to operate, you reduce drastically the cost of your vaccines. And to give you an idea, the one on the right, the 1.65, is the cost of, let's say, very well-known manufacturer for polio vaccines. And you can see that the facility is about 60% or 75% of the cost of the production of vaccines. Buying the facility, operate the facility. You see on the left, all cost modeling, and we have already achieved that. So this is not a modeling; we are doing it. See that the cost of the facility, the orange part, is actually very, very limited. And that actually the USPs of the cell culture and the purification, USP, DSPs, are actually a very, very small part of the production. So, an idea of the evolution. So, starting with a small macrocarrier, 1 to 10 million cells per ml. We have the isolates, which is 100 up to 100 million cells per ml, so 10 to 100 times more, but just the cell culture, not the purification. And with our system, we have an even higher density, and then we integrate the whole system into a continuous process, which basically you feed your cell with food, and you end up with a bag with your vaccine on the other side, mostly automated. So what do we want to do with it? We use that technology to reach our objective, which is making drugs more available and more affordable. So we are basically providing an end-to-end solution to emerging countries for manufacturing their drugs locally. So we basically say to, Brazil or Indonesia or Tunisia, you can build a factory in 2 years, and in 2 years you will make your own vaccines, and you will not have to wait and buy your vaccine on the international market at the same price that America or France is buying your own vaccines. And if there's a shortage, you will not be at the bottom of the pile if you want to buy your vaccines. It works for, of course, any viral vaccine. We are working on that prototype for polio, which is kind of the most urgent needs. We are also working on the same version for therapeutic antibodies and orphan drugs. And of course, thanks to what we call distributed manufacturing, we'll hope to switch from this huge centralized factory that we see for making drugs to small units locally that make drugs for the local people, for in-country, for-country kind of manufacturing. We hope to achieve that kind of objective in the next few years, for at least for vaccines. Thank you.